Before you begin, you'll need the following equipment ready: a 100 milliliter sampling bottle, latex or rubber gloves, a hand towel, a permanent marker, and a cooler or ice container. When sampling for E. coli, always wear gloves and collect from where water is flowing. First, wade into a midpoint in a stream, then wait a moment until the sediment from your footsteps is settled. Fill your sampling bottle facing upstream exactly to the 100 milliliter line and seal the bottle, and use a permanent marker to label it with the site, date, and time. Also, enter the time into your field form. Place the sampling bottle on ice or in a cooler. You have 6 hours to process the sample. Back at the lab or at home, find the Citizen Science E. coli processing mat. Lay it on a flat surface and gather the assets listed on it. And then follow the steps. Heat the incubator to 35 degrees Celsius. Turn on the sealer. Put on your gloves. Write the site, date, and estimated time information on the bubbled sampling tray. Open the media packet and pour its contents into the sampling bottle. Avoid touching or inhaling the powder. Replace the cap and gently rotate the bottle until the media is completely dissolved. Pour the sample into the processing tray, then gently flick or tap the bottom of the tray to release any bubbles. Confirm the sealer is heated, and then align the tray with the holes in the rubber mat and gently feed it into the sealer. Small cells first, white side up. Remove the tray from the sealer and place it into the preheated incubator. Now on the field form, record the time the tray is put into the incubator. And when all your samples are in the incubator, turn the sealer off. Let your samples incubate for 24 hours. After 24 hours of incubation, you will need an incubated sample tray, a Sharpie, the ultraviolet light, and the data form. Remove the sample tray from the incubator and write down the time on the field form. Turn off the lights, hold the ultraviolet light over the sample tray, and use the Sharpie marker to mark the wells that are fluorescing. Turn the lights in the room back on and the ultraviolet light off, then confirm that the wells with the Sharpie marks are also yellow. After this is confirmed, total the number of large wells and the number of small wells that were yellow and fluorescing. Write this on your datasheet. Finally, you can use the IDEQ's MPN table to match up the number of small wells and the number of large wells to get the E. coli results. Write this value on the field form. Submit this data to your Arizona Water Watch coordinator. And after you're done, make sure to dispose of your tray, samples, and other disposables in the red biohazard bag.